Hi, Adipa here. We're going to be doing lecture 2 of Bio 310, DNA replication and repair CRISPR. So here we're going to be focusing on DNA replication, repair, and CRISPR. DNA replication, repair, and CRISPR. So DNA replication is template-driven copying process. We know this because DNA replication always requires template, whereas RNA replication does not really. You have the double-stranded DNA helix, double helix here. Then you have the template S strand, and then you have another template S strand, right? In B, we see that some part of it is breaking out, right? And then in C, we see that incoming nucleotide triphosphates, so the phosphate in the B's are attacking the OH group here. And then something's happening. Let's just read through the process, right? So it's saying that for A, during the process of DNA replication, each complementary strand serves as a template for the synthesis of the new strand. So the cells that start with 2 strands per DNA molecule equals to per chromosome ends up with 4 strands paired in 2 DNA molecules per chromosome. Now, this sounds pretty confusing. I'm going to have this broken down. Um, so essentially, they're stating that you're starting with 2, you're um, ending with 4, right? And these new strands are always synthesized in the 5' to 3' direction. We know that synthesis happens from 5' to 3' direction, whereas addition would be. Okay, my cat was staring dead into my soul. She's right there. That was kind of. I'm scared she's gonna drop my phone. Be right back. Guys, you should say hi to my cat real quick.
She's a kettle. Hmm. Her name's Lily. She's very soft and fluffy and tuxedo, and she's protesting currently.
Yeah, she's gone. Anyways, um, and then the new strand for the same process, the new strand is always synthesized 5' to 3', meaning that the accepting group is 3' on the ribose of the last free nucleotide, and the donor group is the phosphate on the incoming nucleotide. The DNA polymerases are ATPases and hydrolyzes the phosphate bond of the incoming nucleoside triphosphates to obtain the energy needed to catalyze the covalent phosphodiester bond. Now, I'm going to be honest, I need to have this analyzed basically. Summarize easier. So essentially what is happening is, in the first step, there's DNA replication, right? Each original DNA strand is acting as a template to make a new complementary strand. So one is acting basically reproduction, right? One DNA molecule is giving 2 identical DNA molecules. Each DNA molecule has 1 old strand, 1 new strand. So semi-conservative replication. And then B is DNA synthesis direction. So DNA is always made from the 5' to 3' direction. New nucleotides are added in the 3' direction and growing the DNA strand. We know this, right? Synthesized in the 5' to 3' direction, but if you were to add anything, it would be on the 3' end. Easy way to remember this is DNA polymerases can only add to the 3' end, so DNA always grows from 5' to 3'. C is DNA polymerase energy. The DNA polymerase joins the nucleotides together. Um, Together, the incoming nucleotide. I'm gonna share what I have here. I had chat like write up a simpler definition. So here I'm on step C. DNA polymerase energy. DNA polymerase joins the nucleotides together. The incoming nucleotide, DNTP, has 3 phosphates. Breaking off 2 phosphates releases energy. That energy is used to form a new phosphodiester bond between nucleotides. So for what happens first? DNA replication. Then happens the DNA synthesis, where we know that synthesis happens 5' to 3', but you're adding more DNA to the 3' end. That's what's happening. So DNA is always growing from 5' to 3', and then at the end, we are trying to create the phosphodiester bond. How are we creating this phosphodiester bond? We know that there's 3 phosphate three phosphates, right? This incoming nucleotide has three phosphates. If you break off these break the bond between the two phosphates, it's releasing energy. That energy can be used to create this phosphodiester bond between the nucleotides. So essentially, we're breaking to create. So this is a supercharged summary. DNA strand is copied, so DNA replication. Second, DNA we know it's growing from 5 prime to 3 prime, so you're only adding to the 3 prime. And then at the end, you want a phosphodiester bond between the nucleotides. So you break, there's three phosphates, and you break the bond between two phosphates, and you get that ATP in order to create that phosphodiester bond, which would help the nucleotide come together. This allows DNA polymerase to link the nucleotides together. That was a much simpler summary. This is good. Now moving back to here. Original origins of replication and forks. In order for each DNA strand to be copied, the DNA double helix must uncoil and the two strands separate into effectively forming a fork. B. Multiple forks form during DNA replication along DNA molecule. Each fork is an origin of replication. So again, we're going to have this. So origins of replication we see in this diagram, and then the direction of the movement of replication, we're seeing that they have some of it is going up, some of it's going down, it's kind of coming together. And then these are forming replication force, which means it's kind of forming this circular little, you see some circular trends here. If I have this copied onto it's DNA, you have the template DNA, newly synthesized DNA, replication origin, replication forks. Now I'm going to have this here. So you have the DNA unwinding. Before DNA can be copied, the double helix needs to unwind. The two DNA strands separate, forming a replication fork. So first unwind, form replication fork. Then each original strand is used as a template to make a new strand. Origins of replication. The DNA replication starts at many origins of replication along the chromosome, especially in the eukaryotes. Each origin of replication forms a replication fork. Having many replication fork allows the DNA to be copied much faster. So before all the jazz that happened here, right, DNA replication, you need the origin of replication in order for DNA replication to happen. How does this DNA replication happen? How does this fork happen, basically? The fork, the double helix uncoils at first, and then the two DNA strands separate and form a replication fork. So essentially the DNA unzips, fork forms, and then DNA starts copying at multiple origins at the same time, speeding up replication because it's not just one origin of replications, there's multiple. Right, as we can see, there's multiple sites. So these origins of replications are formed. We're gonna talk about coils and supercoils that must be opened and disentangled. So this slide is explaining why topoisomerase is essential for DNA replication. So step A would be the helicase to unwind the DNA. Right, helicase's job is to unwind DNA. A helicase separates the two DNA strands. As it opens the double helix, the DNA ahead of the replication fork becomes tightly twisted. So right here, it becomes tightly twisted, and this creates torsional stress. Okay, we're focusing on A, right? In absence of topoisomers, DNA cannot rapidly rotate and torsional stress is created. So essentially, helicase, you're unwinding the DNA strand, and as it opens, the DNA ahead of the replication fork is becoming like tightly twisted. This is creating some sort of torsional strength. Like right here, it's like twisting. As soon as you open it, it like twists weirdly. You'll notice that with strings as well. If you like, even with hair, if you like there's a breakage and you pull it apart. I have one right here, a split end, right? If you break it apart, you'll kind of see that it coils. Well, I can't do it right now, but it coils. The rest of the thing will coil, and that is creating stress. Think Step B would be supercoil forms. If nothing relieves the stress, the DNA twists into supercoils. It will keep coiling if you don't like untangle it, just like hair. Here's a good example for this. These supercoils make it hard for helicase to keep moving. So DNA replication would eventually stop. So essentially, these coils, you need to stop them, otherwise helicase would stop doing its work. So helicase does its job, separates, and then twisting happens, supercoils. Step C, topoisomerase fixes this problem. Topoisomerase comes in, cuts the DNA strand, one DNA strand temporarily. So it cuts it right here, right? And then this allows the DNA to rotate freely and removing extra twists. The enzyme and then reseals the break. So it's kind of like, yeah, if I break a split end, there's clearly coiling. I would just cut that coiling off, and then it would kind of reseal. The end would reseal. There wouldn't be any what's it called? The split ends anymore, right? It's like resealed back together. So the sequence is that helicase unwinds the DNA, then some coils happen. If it's not relieved, supercoils happen, and helicase eventually has to stop. Then topoisomerase comes in. It's like it temporarily cuts the DNA in half, allows the DNA strands to uncoil, and then the DNA strands reseal themselves. The enzyme then reseals the break. So helicase unwinds the DNA, torsional stress builds, DNA becomes supercoiled, topoisomerase cuts one strand, DNA rotates to remove that stress, topoisomerase reseals the DNA, helicase continues replication. Easy exam summary would be helicase opens the DNA double helix. Problem is the DNA opening the DNA creates torsional strength as supercoils ahead of the replication fork. Topoisomerase makes a temporary break, allows the DNA to rotate, relieve the stress, reseals the DNA, lets helicase continue the replication. Mechanics of DNA synthesis. So these slides cover the entire process of DNA replication. Uh, so you have DNA polymerase, it needs primers, right? A is basically just saying that you need primer to work. It cannot start DNA synthesis without a primer. That's why it's called primer. You're starting with it primes. It needs an RNA primer first. So primase makes a short RNA primer, synthesizes RNA from 5' to 3' direction, gives a DNA polymerase a free 3' hydroxide OH to begin adding DNA nucleotides. So remember, primase makes primer. So you need RNA primer. Primase is making that RNA primer via synthesizing. It's synthesizing RNA 5' to 3'. It gives DNA polymerase a free 3 OH to begin adding DNA nucleotides. Then comes DNA polymerase. DNA polymerase, in the second step, you have that it reads 3' to 5', but synthesizes new DNA. We know that synthesizing, it's always 5' to 3'. Reading is 3' to 5'. It adds nucleotide only to the 3' end. So DNA polymerase always synthesizes DNA 5' to 3'. While copying the 3' to 5' strand straightforward, copying 5' to 3' requires a trick. Primase must synthesize multiple primers on the template strand, and the pieces of DNA synthesized from each primers are called Okazaki fragments. Individuals of Okazaki fragments are ligated very good together by an enzyme called DNA ligase. Leading strand. One strand is easy. DNA polymerase follows helicase constantly. Only one primer is needed. Continuous synthesis. However, lagging, it's discontinuous because it's lagging, right? This strand runs in opposite directions. DNA polymerase must still work 5' to 3', but it also copies little pieces. It needs many RNA primers, many short DNA fragments. These pieces are called Okazaki fragments. Leading is continuous, lagging is discontinuous. So what happens to the RNA primers once they do their job, they have prompted DNA polymerases to start working, right? If after DNA is copied, the RNA primers are removed. DNA polymerase fills the gaps with DNA. DNA tiny breaks remain. The DNA ligase joins everything together. Think ligase as a glue, it seals the sugar phosphate backbone.